সেল এক জায়গায়, ডিস্টর্টেড কিছু নেই, দুটো তিনটে সেল এক জায়গায় চলে এসছে। যত ভালো এ হবে তত ইউনিফর্ম, এটা ভালোই আছে, কাউন্ট করা যাবে। এটা তো হাই পাওয়ারে আছে, তো 40-তে আছে। ওই স্কোয়ার গুলো কাউন্ট করবো, যে স্কোয়ার গুলো রেডে মার্ক করা আছে। আগে তো স্কোয়ার আইডেন্টিফাই করতে হবে, হাই পাওয়ারে স্কোয়ার আইডেন্টিফাই করে করতে হবে। টপ লেফট ওই অনেক সেল, অত তো আসার কথা নয়। মোটামুটি অ্যারাউন্ড 1 স্কোয়ারে মোটামুটি 100টা থেকে 120-130টা এলে তাহলে ঠিক আছে। ব্যাক ক্যালকুলেশন করলে তাই পাওয়া যাবে। প্রচুর আর্টিফ্যাক্ট আছে, ঠিক আছে।
মানে এল শেপে যে কাউন্ট করতে হয়, কি এই লাইনগুলো কাউন্ট করব?
যদি একটা সিঙ্গেল স্কোয়ার কাউন্ট করার সময় এল শেপ মানেই তো এই লাইনটা, এই দুটো বাদ দিতে হয়, ওই দুটো বাদ দিতে হয় যাতে সেল ডবল কাউন্টিং না হয়।
না না স্যার, ওটা বলছি না, এই যে তিনটে লাইন না, তো কি তিনটে লাইনে যেগুলো আছে সেগুলো বলবো না?
First you have to do it properly, then calculate also properly. Where is your book? You have been told from the first day to carry a practical textbook, at least for one group, one book. If you are 3 people, carry at least one book. What will you refer to? Can you do it by yourself? If you are confident, then do it. After 30 minutes, I will see your results. Where is your textbook? How many in your group? One is absent. Okay, 3. So one textbook is to be carried. What will you refer to if you can't follow a step or if you forget the calculation part? This is a tough topic. There is some, you have to do it properly. When you have to see it under the low power Neubauer chamber, how to focus. There are different steps, and then you have to calculate. Calculation part is also for the beginners. You have to understand. So what will you refer to? There is no use just coming and standing and doing one part and going away. If you have a problem, what will you refer to? You should carry the textbook. How many people? Why 4? We are giving at least, we have 16 tables over here at least. And we are encouraging for 3 people, 50 people, 3 people in one group. Why are you 4? 1 if you join them. After roll numbers are allotted, it will be strictly roll number 1, 2, 3, 4, 5, 6, maximum 3 in one group. Two in this group, two persons. And in your group, how many? 2. Where are your textbooks? Textbook. What practical textbook? What will you refer to if you find some difficulty? There are so many steps in this experiment. There are calculations. From the first day, we are encouraging that bring one textbook. For one group, at least one textbook. Without textbook, it is, you will do half of the thing. For 50 students, there are 1 or 2 faculties you can't learn like this. That what is focused over there is only the squares to help you, only the squares. How will you calculate? Do you know how to calculate? If you know, if you are confident, do. I'll see your result after 30 minutes. We have already told carry the textbook. Otherwise, you will do half the thing and go away. If you have studied and come, already you can perform, you are confident, you can perform without the textbook as it is in the exam. What you have to do in the exam. In the first or second class, it is good enough. Then do it and show. What's the name of this fluid? What's the name of this fluid?
Heme fluid.
Hemocytometer fluid. What does it contain? Sodium chloride. Right. What are the functions of the components? What are we doing by rubbing the bulb like this between your hands? What are you actually doing?
Mixing the heme fluid with the blood.
Yes. What is the purpose of that? Obviously, mixing the heme fluid and the blood. What is the purpose of that? So refer to your textbook when you are waiting. Understand the topic and do it. What does sodium sulfate do here? Blood can be diluted even with normal saline, even with water. If we give tap water, there will be impure materials. You can do it with even with distilled water. Then why are we using heme fluid? So you are MBBS students. Understand and do scientifically. Mercury chloride is given for preservative. So if the solution is freshly prepared, mercury chloride is not an essential component. But sodium sulfate and sodium chloride are required. Osmolarity, that is sodium chloride, and sodium sulfate. The word is not Rulex, the word is Rulo. RBC already taught in theory class. Word is Rulo formation. Word means the pronunciation is Rulo. It is not Rulex. Yes, that is the purpose of sodium chloride. So that is, now by mixing the RBC with the fluid, what are we doing? First, we are preventing the Rulo formation. Otherwise, what is Rulo formation? RBCs get stacked up. If they are stacked up, we cannot count them. So that is the purpose of doing that. First purpose, and then mixing so that uniform distribution is there. If already is mixed, what is the next step? After mixing, what we have to discard? Which part have you discarded? After mixing, you have to discard. What is in the step? Mixing is occurring here. In this part, there is no blood. There is only fluid. So that will not. If we charge that, then that is not participating in cell counting. That part has to be discarded. So discard that. Then you have to charge. So what to do if there are bubbles? There is no other option than to do it freshly. Otherwise, you will not get the result. Bubbles should not be formed. If bubbles are formed, how actually what you are thinking to be, what you are taking to be fluid, that is actually air. Then counts will be erroneous. Out of 100 parts, if 20-30 parts are bubbles, so actually that is 70 parts, then 70 parts of volume. Every step is important. Even if you can discard the bubbles, how will you get the volume, actual volume? Not possible. How do I identify the RBC pipette? Which is the RBC pipette? in this total apparatus, which is the RBC pipette. This one, yes. How do you identify this? What is the, that means identification means you have to differentiate this from this one. This one is the WBC pipette. So how do you differentiate? Because of this, which, because of this portion, what characteristic of this portion helps you to differentiate between this and this? This is a larger. Okay, that is one point. First point is, you see the markings. Markings are given like this. This is a markings. What is the difference in marking? In the lower part of the stem, markings are same, but here the marking is 101, here the marking is 11. And the bulb is larger, the bulb is smaller. Those are the two differentiating points. Name of the counting chamber? It is named after the person who described it first, who invented it. So Neubauer's chamber, but it is called the improved Neubauer's chamber. Now after thing else, now it is called the improved Neubauer's chamber. Why it is called improved Neubauer's chamber? Read the book and tell me. If the pipette is okay, why will it go away? The pipette is made like that. What is the composition of this fluid? What is the name of this fluid? Okay, you do the work. Tell me what is the name of this fluid? Hems fluid. Hems fluid. The pronunciation is Hems. What are the components of Hems fluid? Why are you using this? Better to dilute. If you don't know, then understand everything and do the practical work. If you don't understand, what is the use of doing this? Why are you using the Hems fluid? Dilution can be done simply with distilled water. Why are we diluting the RBCs? First of all, why are we diluting the blood? Don't dilute the RBCs. Why are we diluting the blood? We could count without diluting also. Then what would happen? RBCs number are already large. So so many cells cannot be counted together in a small area. That is the purpose of dilution. But why diluting with the Hems fluid? Why not with distilled water? Mercury chloride. Okay. That you have memorized. But what is the, why are we, okay, we do the work and tell. Have you done? Have you taken blood? Have you already used this today? Have you taken blood and mixed with the fluid? Okay, if bubbles are there, actually the calculation will be wrong, but still you can do, but bubbles should not be formed. Along with doing the experiment, read the book and at the end of the class answer. Why not with distilled water? Why should we dilute and why the Turk's fluid is used? Sorry, Hems fluid is used. Starch fluid is for WBC. We will do next week.
Clotting or Rolex formation.
Clotting and Rolex formation is not the same. Rolex, the word is rouleau. Okay, the word is a French word, rouleau, not Rolex. The pronunciation is rouleau. But rouleau formation and clotting are not the same. Blood clotting is something else. That is blood coagulation. So, okay, rouleau formation. So, so if there is rouleau formation, what does rouleau formation means? Rouleau formation is not clotting. What does rouleau formation mean? What is it? Stacking of RBCs together.
Stacking or RBC.
That is to be prevented. So there has to be something that prevents that. So Hems fluid components are such that it helps to maintain the osmolarity, it helps to prevent the rouleau formation, and also mercury chloride, if it is a fresh solution, may not be needed, but if it is a stored solution, it is a preservative, antibacterial and antifungal. So that is the purpose of mixing, and also the purpose of mixing in the bead is uniformly spreading the cells. They make a uniform solution.
To prevent clump formation of RBC বললে হবে যদি Rolex formation নাম বলতে চাই।
না চাই কি rouleau formation, the word is rouleau formation. What have you? So before doing this thing, you should focus already on the low power. Because you have to charge. Before the cell, after uniformly distributing the cells, you should charge quickly. So focusing should be done earlier, not later after. What are you doing? Then you should charge. Waiting 5 minutes. Okay, 2 to 3 minutes will be enough. But have you focused? First, focus is going off.
I'm focusing, but it's getting defocused.
No, it's a good focus.
But I don't know, sometimes it's becoming hazy.
No, it's okay. Even at this point, it's okay. Now this is the RBC square, right? The central. So you have to now focus which is the RBC square, and then at the time of counting, you have to do it under the high power and identify the squares. This is okay. This is which square? No. 45x.